Hello friends, welcome back to my channel, and today in this video we are going to study about B cell signalings. We are studying the signaling pathways from quite few videos, so nothing new in this pathway, just it is a summarize or it's a summary for you to understand that among all the signaling pathways, which signaling pathways come under specifically B cell. So in that, the first one is calcium signaling pathway, or we can also say it as calmodulin calcineurin pathway. So what happens in this pathway? We already learned PLC gamma 1, that is phospholipase C gamma 1. It degrades PIP2, that is phosphoinositol biphosphate, into DAG diacylglycerol and IP3. IP3, the molecule, it have the receptor in endoplasmic reticulum. Once it binds with the receptor present on the surface of endoplasmic reticulums, in return, in response to that, endoplasmic reticulum releases calcium. This calcium will binds with calmodulin and make it active to for bind with the calcineurin. And how many calcium molecules are required for this? 4. So once calcineurin binds, calcineurin acts as a dephosphatase. That means it removes phosphate. So NF80, which is a transcription factor, but it remains inactivated due to this phosphate attached with it. So calcineurin will remove this phosphate. Hence, NF80 becomes activated. It can cross the nuclear membrane and can function as a transcription factor for B expression of the particular gene. Next pathway is from the DAG, which is produced RAS GRP bind SOS. It is a guanine exchange factor which replace GDP with GTP. Now the RAS GTP is activated. It will phosphorylate RAF and hence the RAF become activates. The phosphorylated RAF will phosphorylate MEK, making it active. MEK will in turn phosphorylates ERK. ERK can enter the nuclear membrane and activates ELK by phosphorylating it. ELK and SRF helps in the expression of the gene which is known as FOS. Now gene gets expressed but still it is inactive. So this ERK, it phosphorylates the FOS, making it active. Once the FOS becomes phosphorylated and active, now its partner Jun will come and bind with it. Now this is a complex active form which is Jun FOS complex. Now this can activate AP1, which can act as a transcription factor for the expression of the genes. Now the third pathway from the DAG itself, it can activate TAK1 complex and KARMA1 complex. These two will helps to activate IK, K alpha and beta, which helps to break I kappa beta. Once it is broken, now NF kappa beta becomes active. Now it can cross the nuclear membrane and acts as a transcription factor for the expression of the gene. So these are the three main pathways involved in B cell signaling. Now this is the summary of all the pathways, including, as you can see, this is the calcium pathway, this is the nuclear factor kappa pathway, and this is the AP1 pathway. Now apart from that, because the expression of these genes, they release cytokines. So for the release of cytokines, the necessary is cytoskeleton reorganization, because the position where the cytokine is getting released is important. Say for example, if this is the cell and here cytokines are present. Now for the release of cytokine at this particular location, it has to move up to here. If this is the position where cytokine is to be released, then this cytokine filled vesicle should move up to here. So this is done by cytoskeleton reorganization. Now apart from that, all these expression are for the proliferation of the cell. Correct. So one more pathway is needed for the proliferation of the cell is anti-apoptotic pathway. That means to stop the cell death. For that, the PIP3 with it AKT and PDK1 binds, that helps to inactivate BAX and BAD. So these will stop the apoptotic pathway and hence the cell will move towards the survival pathway. So this is all about B cell signaling, all the pathways that are involved in the B cell signaling. So that's all for this video. If you found the video informative, please do like, share and subscribe the channel and hit the bell icon for the next video notification. Till then, stay safe, stay happy.